So restriction enzymes are available that recognize a wide variety of sequences. Most of them recognize sequences between 4 and 8 bases in length, and you can see in this table several sequences recognized by restriction enzymes. Another common feature of restriction enzymes is that they recognize symmetric sequences, meaning that the sequence that reads 5' to 3' on the top strand is the same as the sequence that reads 5' to 3' on the bottom strand, meaning that they are what we call complementary sequences, and that both strands get cut by the restriction enzyme as a consequence of its action. Now, you can see the action of an enzyme called HinD3 acting to cut a double-stranded DNA here. And because of the staggered nature of the cuts, one ends up with DNAs that have what we call sticky ends. And so here's an example of an enzyme that recognizes the sequence CTG-CAG. It's the enzyme known as PST1. And you can see that it leaves overhangs, that is, sequences that aren't flush with each end, but that there are overhangs onto those. Those overhangs can be places for overhangs of other DNAs to be linked up with it, and that's actually used in the process of making recombinant DNAs. Yes. So the recombinant DNA is made by, first of all, taking a sequence known as taking a DNA known as a plasmid. A plasmid is simply a circular DNA that's found in a bacterial cell that'll replicate in a bacterial cell. I can cut that plasmid at a specific point, as you see has happened over here. And if I don't do anything else, I just simply ligate it back together using an enzyme called DNA ligase, I'll just recreate the original plasmid that I had. However, if I take and I put other DNAs in there using those overlaps that I talked about, I've now joined these guys down here with the plasmid sequences. I've recombined, I've put things together, and I've made a recombinant DNA. So when you hear about recombinant DNA, that's how it's made. It involves starting with a restriction enzyme and a plasmid, and then linking things together, as you can see here. There are many different plasmids that are available for researchers. Plasmids are typically relatively small DNAs compared to a chromosome. So an E. coli chromosome, for example, might have 6 million base pairs. It would be unwieldy for us to work with in the laboratory. Plasmids, on the other hand, have sequences. This one has a sequence of about 2,800 base pairs and is much more manageable. The larger a DNA is, the harder it is to keep from breaking it, for example. So something like this is very easy to manipulate and relatively easy to put into cells. If I use this plasmid, I could cut this plasmid at a variety of sequences that you can see, marked by the individual sites that are on here, to create recombinant DNAs.